All right. All right, let's get going. Yeah, so it's wonderful to be here. Thank you for the invitation. I want to tell you a bit about the kinds of ideas that we've been excited about in my group in the physics department at MIT over the last 5 years. So broadly, we're interested in trying to understand how interactions between members of a population kind of dictate either the evolutionary or the ecological dynamics of the population. And in particular, what we do is we use experimental microbial populations in the lab in order to kind of explore some of these ideas in theoretical ecology. So I'll give you kind of the main stopping points over the next 45 minutes or whatnot. Right, so it turns out that the natural populations can collapse suddenly in response to deteriorating environmental conditions. But from some ideas from nonlinear dynamics, there's at least, in principle, some universal behavior of these populations near such tipping points. And it may be the case that that universal behavior may allow one to, in some ways, get some advance warning that transition is about to take place. And at least in the laboratory, we're able to measure some of these theoretically proposed indicators, such as a change in the fluctuations of the population size before a collapse. Now, the basic reason that these populations can collapse suddenly is because of some positive interactions between the members of the population. Right, so if there's positive feedback within the population, then below some size or density, the population may not be able to sustain itself, and then you can get this catastrophic collapse. So it's known as the alley effect in ecology. Right. So anytime you have such cooperative behaviors, you have to ask, well, what happens if some member of the population doesn't contribute to that public good? And what we find experimentally is that such cheater strategies can indeed spread in the population. Right. So somehow that state of cooperation is unstable in an evolutionary sense. All right, and then finally, if there's time, I'll tell you a bit about some of the population-level consequences of these cheaters in the population. All right, so the way that we often think about this is by a bifurcation diagram. So what I'm plotting here on the y-axis is the size of the population in response to some environmental condition with deterioration on your right. All right, so what can happen for the case of, say, a fishery that collapses suddenly is that as the environment deteriorates, so for example, increased fishing pressure, it can happen that the size of the population decreases, but only in a modest way. And however, at some critical level of the environment, some critical level of fishing, what can happen is there can be a catastrophic collapse of that population. Right? So this could be local extinction or transition to some other, for now, we'll say undesirable state, like a eutrophication of a lake. Okay. Right, now, okay, so this happened, and you say, oh, well, that was really, that was bad. Let's just fix it. And you'd say, oh, we can just improve the quality of the environment. However, things may not be so simple because in the presence of those feedback loops, it may be that there's a hysteresis or memory in the system. Right? So it may be the case you have to come all the way over here to the left before you can get recovery of the population. Right? So what's happening here is that there are these two stable states denoted by the solid line, and those two stable states are going to be separated by some unstable state, right? This dashed line. Right? So that unstable fixed point, that's telling you about the minimal size of the population required for survival in some particular environmental condition. Right? So if you're above it, then you'll come to this state, whereas if you're below it, you'll come down here. Okay? All right, so this is known as a fold bifurcation here. And it's the basic mechanism that's thought to lead to sudden transitions in a wide variety of complex systems. So obviously, we're interested in this in the context of populations and ecosystems, but other researchers are thinking about these dynamics in the context of epileptic seizures, climate regime shifts, financial market collapses or tipping points. I guess the other conference here might be thinking about such things. Right, but in all these applications, what you'd really like to know is whether there's any way that you could have anticipated that this transition or tipping point was about to come. Right? Because absent really detailed knowledge of the dynamics of the system, you don't know that it's going to happen here. Because it could be that it occurs over here, or maybe it's just that the population size smoothly goes to 0 in response to a deteriorating condition. Right? So what you need to know is some way to anticipate that you're kind of right here without really detailed knowledge of the dynamics of the system. And in principle, one might think that is even in principle kind of impossible. But what's interesting is that from nonlinear dynamics, there are some, at least theoretically proposed, universal indicators that a system is approaching one of these bifurcation points. Right? And to get an idea of why that might be, what we can do is we can take that bifurcation diagram, and we can take kind of two slices of it. So the green slice is far from the tipping point here, F2, so that's the bifurcation. The red slice is when we're very close to it. Okay? Now, what we're going to do is we're going to kind of rotate things. So now it's going to be the population size that is on the x-axis. And instead, we're going to look at some effective potential on the y-axis. It's going to tell us about how the population or the system responds to perturbations. So in this green slice, when we're very far from the tipping point, the effective potential is kind of very nice and steep walls. Right? So this is telling us that this population right here, it's somehow in a very stable state. Right? Any perturbation away from equilibrium quickly goes away. Moreover, there's a very nice, large basin of attraction. So a perturbation would have to be very large in order to push the system past this point here in order to induce a transition to this alternative state. Okay. However, as we move towards this tipping point, so as the environment deteriorates, what you see is that the effective potential looks very different, right? Because now this potential is really very broad. And this is saying that a perturbation away from equilibrium is going to take a long time to go away. So there's a decrease in the rate of recovery to a perturbation. Moreover, that decrease in the rate of recovery, it manifests itself in the fluctuations of the population, even in the absence of a defined perturbation, just because there are going to be some sort of fluctuations in the population, whether small environmental fluctuations or demographic fluctuations. What that means is that you expect a change in the fluctuations of the population size when we're close to the tipping point here as compared to far away. And in particular, the expectation from theory is that the fluctuations should get both larger and slower. And finally, you can see that this basin of attraction is also shrinking as the environment deteriorates. So the population you expect to therefore lose resilience to perturbations because the basin is shrinking. Okay. All right, so these ideas result from this phenomenon of critical slowing down, which some of you may be familiar with. And the phenomenon is actually in some ways more general than what is indicated in this slide here because it occurs even when the dynamics of the system cannot be described by an effective potential. Right? So in higher dimensional systems, it's often the case that the dynamics are more complex, but the general statement is that at these sorts of tipping points, the dominant eigenvalue of the system goes to 0, and that results in these signatures of an impending collapse. All right, so I just want to summarize kind of what's expected from theory here, which is that as we approach one of these tipping points, we expect there should be a loss of resilience to perturbations. But that loss of resilience, maybe we can anticipate that it's occurring based on some other signatures within the population. In particular, there may be these early warning signals, an increase in the recovery time following a perturbation, and also an increase in both the size and the timescale of fluctuations in the population size. Okay. If there are any questions, now is actually a fine time just to make sure we're all talking about the same thing before getting into the experiments, measurements. That's fine. Don't feel.
So the second one you can use if you had a real population, if you were observing some.
Okay, right. In principle, both of these could be used, right? Because the idea is that, well, you could perturb it yourself. Well, that's not always advisable, right?
Looking at some bear population.
Right, but right. So the idea is that such a perturbation could be a drought, or it could be, the idea is that the environment every now and then does have these sudden kicks, and then you can look at how the population recovers, or you could perturb it yourself. But it's true that for this one, the increase in the recovery time, there has to be this defined perturbation. Whereas in the second case, you can just, in principle, look at the fluctuations. Yeah.
There's one thing I do not entirely get. I do get that the fluctuations get slower, but why would they get larger? Because your basin of attraction actually gets smaller if you come closer.
Okay, so the basin of attraction gets smaller, exactly. Right, so the statement is that you expect the fluctuations to grow, and that's because in all these cases, we're thinking about the dynamics somehow near equilibrium. So we're thinking about kind of the small noise regime when we're not actually getting transitions yet. Okay. So in that sense, the size of the basin of attraction isn't yet relevant for this question. Okay, so instead, right, just natural demographic fluctuations, for example, they're always hitting the system. And if it's taking longer for those perturbations to go away, then they kind of add up on each other. And then that results in a larger kind of net size of fluctuations.
I mean, how general is that kind of statement? Surely could there not be a system that just has a different spectrum? Or
Okay, so that many obviously this is a large field where many people have been studying cases where this basic phenomenon is going to fail, but in many of those cases they're somehow mathematically degenerate in the sense that the potential has a sharp kink, or if you introduce something that is kind of funny, then you can get this. But because the mathematical statement is very general, right? A fold bifurcation is a zero eigenvalue bifurcation, right? So there's that's a mathematical statement rather than a statement about my yeast populations, right? But it's worth saying that so the bifurcations that many of us are familiar with, the local bifurcations, this is a fold bifurcation or transcritical or hop. These all are zero eigenvalue bifurcations where this critical slowing down phenomenon is expected to occur. Whereas some of the more global bifurcations, it won't necessarily happen. Yeah. All right. Okay, so I think that these are really interesting theoretical ideas, but from my standpoint as an experimental physicist, our goal is to kind of take some of these, be motivated by these theoretical ideas, but then go make real measurements on some real system. So what two students of mine decided to do is to try to measure these phenomena in laboratory yeast populations. Okay. Now, what you need to know about our yeast right now is that, okay, so yeast, single-celled organisms, few microns in size, and they're wonderful from the standpoint of experiments because they're small, simple, they divide every few hours. We can perform quantitative measurements of things like population size. We can perform the perturbations ourselves. We can measure fluctuations. So we can do kind of everything that you would like to do with fisheries, but we can do in the lab here with our yeast that you don't necessarily care about a priori, but they're at least a nice test bed for exploring these ideas of theoretical ecology, right? Okay, so what you need to know about these yeast cells is that when they're growing on the sugar sucrose, they somehow grow cooperatively, okay? So sucrose is a disaccharide that needs to be broken down in order to be consumed, and that breakdown occurs outside of the cell. Okay? So the yeast, they secrete the enzyme that breaks down that sugar, and that leads to this cooperative growth, and in particular this alley effect that many of you are familiar with. And what we can measure just experimentally is that the yeast actually divide more rapidly at higher cell density. Okay, so it's not just that the division rate goes down, down, down with density, but instead, over some regional densities, it actually kind of first goes up and then later goes down. All right? So there's some ways in which those yeast cells are benefiting each other. Okay? And it's that dynamic that at least the possibility of sudden collapse, but of course, we have to measure to see if this is what happens. Okay?
Do all cells express the gene?
Yes, in this case, they're all, and we even have single cell reporters to confirm this is the case. All right, so these experiments are rather simple, so I want to make sure that we're all imagining the same thing. What we do is we're growing our yeast in well-mixed liquid batch culture. And what that means is that we start with some low density of cells, and we allow them to divide over the course of a day as things are being kind of well stirred, or shaken rather. All right, so at the end of the day, we can measure the cell density just by looking to see how cloudy that liquid is, the optical density. All right, now that's going to be the population size or density that we're going to be plotting in the later figures. And what we do now is we perform just a daily dilution where we transfer a small fraction of the cells into fresh media with fresh nutrients, including sucrose, and we repeat this process for a week or two. All right, now, we're going to control the quality of the environment, at least initially, by varying this daily dilution, which is equivalent to some sort of mortality rate that's imposed on the system. Okay? And in particular, right now, we're going to start with a dilution of 1,400. So what that means is that only 1 in 1,400 of the cells that are present at the end of this day will be allowed to propagate into the next day. Okay? So the first thing we wanted to do is ask whether it's really the case that yeast growing on the sugar sucrose display bistable fates depending upon the starting cell density. Right? Very difficult thing to confirm is happening in the context of eutrophication in a lake or in a fishery or whatnot, but in these populations it's really simple. Right? We just start at different cell densities and then maintain them in a constant environment and see what happens. And when we did that experiment, what we found is very clear evidence of bistability. Right? So many different population densities over orders of magnitude in size. All right, propagated constant environmental condition over time, dilutions each day. What you see is that if we start with a large cell density, those populations all reach some stable equilibrium population size. If we start with a low density of cells, those populations are all shrinking over time. So they're going extinct, and we can confirm that independently. Right? So, and it's important to mention that it's not that the cells are unable to divide in this environment, but rather we've started the populations at lower cell densities, so there just are not enough cells to break down enough of that sucrose to allow the population to divide enough times to counteract this daily death process, this dilution. Okay? Right, and between those two stable states, you expect there to be an unstable state, and indeed we can see kind of directly where that is. Whereas here, these 8 populations, although started at the same cell density, what you see is they're experiencing stochastic fates, and that's because we've placed those 8 populations on the top of that effective potential. And you can see that it's taking a long time for the fate of those populations to resolve, and something like half of them look like, okay, they may survive, but half are going extinct. Right? So we can see quite clearly where that unstable fixed point is. Okay? So that's telling us about the minimal population size or density required for survival of the population. Okay? All right, so what we can do now is we can take, at this environmental condition of 1,400, we can plot the locations of those fixed points. Right? We can plot, okay, here's the stable population size / density. Here's the minimal size required for survival, and again, the stable state down at 0. Okay? That's at a fixed environmental condition, but what we want to know is whether this population will really experience one of those fold bifurcations that is going to lead to a sudden kind of population collapse, right? In order to get at that, what we can do is we can repeat that previous experiment, but for many different qualities of environment, many different dilution factors. Okay? And indeed, when my students did that, what they found is they got a really beautiful fold bifurcation describing how yeast grow on the sugar sucrose. And it has the features that you kind of expect from those from the simple theory. The size of the population indeed shrinks as the environment deteriorates. However, the minimal population size required for survival, that grows, as you would expect. And indeed, the tipping point, or the bifurcation, it occurs over here where the stable and the unstable branches collide and they annihilate. Right? And that's what leads to this catastrophic collapse, leading to extinction of that population. Okay? All right, so I think that this is a really important place to start when studying these sorts of ideas, because in this field, many researchers have a model suggesting that their population should have a fold bifurcation. But in some ways, you can always write down a model that gives you any feature that you want. If you don't have experimental data to demonstrate that's what's going on, then you're in a little bit of shaky ground, right? So in this case, this is actually the first case that I'm aware of where it's actually possible in a biological population to experimentally map out a fold bifurcation and demonstrate that's what's going on. And then you can go and you can test all these ideas in theoretical ecology, right? So for example, we can test this basic idea of a loss of resilience approaching this tipping point. Okay, so resilience, once again, is defined as the ability to withstand perturbations, right? And this kind of makes sense because, again, the basin of attraction is somehow the distance between the stable and unstable states here. So a given perturbation on this population will push the population here, but you can get recovery. Whereas the same perturbation on these populations may push them below that unstable branch and then will lead to extinction or collapse of the population, right? So that's why you kind of expect a loss of resilience quite generally as one approaches a tipping point. Yes?
Does the position at which the tipping point occurs depend on the total size of the population? Or only on the...
No, so it's so it really depends on just on the quality of the environment, because you rapidly or relatively rapidly come to this state. And of course, if you start up here, then
no, I'm asking about the total size. So you're always giving us numbers.
oh yeah, no, it's really density is the relevant thing. So if you had a larger volume, then indeed
So demographic fluctuations are, they always will give you some correction, but you are saying
right. Those are small, right? So yeah, and indeed, in general, in our populations, the root n or demographic fluctuations are not the dominant source of noise, but it's really more a linear n noise. Right. Okay. All right. So let's okay. So we can go and we can explore this idea of a loss of resilience. So what we can do is we can take populations, allow them to reach equilibrium at different environmental conditions, and then just perform a perturbation. In this case, what we did is we just over one day we grew them in a bit of salt, all right, or higher salt concentration, so that now what you can see is that okay, these populations they're at equilibrium, but they're in a very benign condition. So with this salt shock, the population size goes down, but then when we return to the original environment, we get rapid recovery. Okay? Now, if we go to a more challenging environment, now there's a larger dip in the size of the population, but it's also taking longer to recover from that perturbation. Right? So we can already directly, in these populations, see this basic phenomenon of critical slowing down, an increase in the time to recover from perturbations as the environment deteriorates, as we approach a tipping point. In a more challenging environment, now what you can see is that some of those populations may survive, but some may not. It's really right around that unstable fixed point. In a more challenging environment, now you can see that those populations are all going extinct. Okay? And this is all true despite the fact that if we had not perturbed the populations, all these populations would have survived. Right? So this is really mapping out the loss of resilience to a perturbation as the environment deteriorates. Okay. And it's worth mentioning that Martin Scheffer in the Netherlands measured this increase in recovery time following perturbation in another microbial system that was in continuous culture in a chemostat. Right? So there's no sense that this daily dilution procedure that we use is leading to any of these results that we're talking about. Okay. All right, so now we can come back to our bifurcation diagram and we can ask, all right, well, we know that there's going to be a loss of resilience to perturbations, and we've seen. So what you'd want is some sort of indicator that's telling you that you're approaching this tipping point and is telling you that your system is going to be losing resilience, right? And we should be able to do that by looking at the fluctuations as well as the increase in recovery time. And indeed, what we can do is we can measure the magnitude of the fluctuations by looking at the coefficient of variation. So this is the standard deviation divided by the mean, and this is measured over roughly 20 populations over 5 days. Give you a sense of kind of the amount of data that's coming into this. So what you can see is that in a benign condition, the fluctuations are really quite small, just a couple percent. Right? However, as the environment deteriorates, as we approach this tipping point, we see really quite a dramatic increase in the size of the fluctuations in this population. Right? So this is consistent with the predictions from critical slowing down, and also is telling us that we could then use this coefficient of variation as an early warning indicator that the population is approaching some sort of tipping point and that we have to intervene to save our yeast populations. Now, I think given the nature of our noise, this linear noise and so forth, I think the coefficient of variation is the kind of the proper metric for noise here. But it's worth mentioning that even if you just look at the standard deviation in those populations, right? So just the raw magnitude of fluctuations, even that's actually growing as the environment's deteriorating. Right? So despite the fact that the population size is getting smaller and smaller, the fluctuations are actually growing. Okay? So this is not what you'd expect from any other kind of model of noise. Okay? All right, so this is saying that the size of the fluctuations really can serve as some sort of early warning indicator of an impending tipping point. And indeed, we also measured an increase in the autocorrelation time, or the time scale of these fluctuations. Right. Did you have a question?
No, I was just wondering how the fluctuations look like, because when we see the raw data, it's almost a flat line.
Right, yeah. Right, so maybe, I do have the raw data somewhere here. I'd have to, maybe I'll find you later and I'll show you what it'll take me a few minutes to find it.
The really small scale fluctuations around the mean, right?
Well, you can see here that the fluctuations are, maybe as we're getting closer, maybe something like 10%, right? And I'd say that we can't actually measure anything lower than this because our measurement error is around 1%. So we're really, this is very close to just measurement error. But these things, 10% is a fair size. All right. Now, I just wanted to tell you a bit about some of the things that we've been also thinking about very recently, and we're kind of preparing a manuscript on, which is the question of how you might try to compare the strength or the performance of these early warning indicators in different environmental conditions. Right, so part of the allure of these early warning indicators based on critical slowing down is the idea that they're somehow universal. Right? They should occur in my yeast populations, they maybe should occur in fish populations, but also within a given population, they should occur regardless of how the environment is deteriorated. Right? In our case, we increase the mortality rate, but what if we did something else? What if we decreased, for example, the concentration of the sugar that's available to the population? Right? So we did that experiment, and first we did it in kind of continuously deteriorating environment, where we deteriorated it over the course of 3, 4, 5 weeks. And again, when we saw these indicators, but then we said, oh, well, it seems like they're not as strong in this sucrose condition as in this dilution factor condition. And we said, oh, well, what's going on? We thought, oh, maybe it's because we're deteriorating the environment maybe too rapidly in the sucrose condition. Okay? So then we went and we just fixed the environmental condition. And then we measured, for example, the coefficient of variation as a function of sucrose concentration, coefficient of variation in this dilution factor. And you say, oh, well, yeah, so the coefficient of variation goes up more over here in this dilution as we deteriorate the environment by increasing dilution factor as compared to the sucrose condition. But then after you make that statement, you realize that it's not a very well-posed question to begin with, because as you get closer to the bifurcation, all these things are supposed to go up, right? So maybe it's just we haven't gone close enough in the sucrose condition, right? And the fact is, you can't compare things at the same distance to the bifurcation because these aren't even the same units, right? This is in units of sucrose concentrations. This is in units of dilution factor. So you're kind of just stuck here. And indeed, we made some of these original measurements a couple of years ago, but we didn't know what to do with them, right? Because we didn't know how to compare the quality of the indicators in these two different environments. And it was only somewhat more recently that we realized that what we should be doing is we should be comparing the indicators in terms of the strength indicator holding resilience constant. Resilience is this ability to withstand perturbations, and in many cases, populations, they don't go extinct because you actually reach that bifurcation point, but rather you go extinct somewhere before that because some environmental perturbation is going to push you over the edge and lead to extinction. So what you want is to be able to see a rise in these indicators before resilience gets too low. Okay? And what's great about this is it allows us to plot the strength of these indicators on the same x-axis, because now we have a unit that is resilience that is common between the two. Right? And indeed, we can then go, in order to measure the resilience, we can map out the bifurcation diagram. Okay, again, you've already seen this. This is the bifurcation diagram in the dilution factor condition. But we measure the bifurcation diagram in this sucrose condition as well. It's, again, a nice fold bifurcation, except now the tipping point occurs over here on the left because we're decreasing sucrose concentration. Right? So, but now we can actually plot these indicators on the same axis since we've measured these things. And indeed, what we find is that the coefficient of variation indeed goes up more in this dilution factor condition than in the sucrose condition, at the same value of the resilience. Right? So we think that this is indeed maybe a nice way to compare the performance of these early warning indicators in different environmental deteriorations. Okay? Now, the basic underlying reason that this might happen, I think we can get a lot of kind of conceptual insight by thinking about the path of the environmental deterioration in some sort of landscape of resilience and stability. Because stability was this thing about kind of the local shape of the potential, right? And it's as the potential kind of broadens, that is indeed what leads to all of these early warning indicators associated with critical slowing down, right? So there's a loss of stability as one approaches the tipping point. And so the way that we're thinking about this is that if we start out over here in some kind of good environment, we know that this has some reasonable resilience and high stability. Okay, so it's a good state, and we know that eventually, as you deteriorate the environment, you reach the origin here where you've completely lost stability and you've lost resilience. Okay. So our argument is that depending upon the different environmental drivers, there's going to be a different path on this stability resilience diagram. And indeed, we were able to measure this experimentally. But the statement is that we want to be able to see the loss of stability before we get too low in resilience. So this is saying that this path here, driver 1, actually will lead to a better performance of these indicators as compared to this the sort of magenta or purple line associated with driver 2. So we think that somehow different environmental deteriorations will lead to different paths on this space of stability and resilience, and that's what's going to dictate how well the early warning indicators work. Okay. Are there any questions about what I just tried to explain?
Would it make sense to define something like the temperature small t that you will define in statistical physics once you talk about transitions?
Okay, right. So this actually gets to this question of the other way that you could try to compare these indicators is instead of looking at the resilience, you could try to look at the lifetime of the state, which is related to this some effective temperature, right? And the lifetime of the state is actually then a function of both the stability and the resilience. So that would be another way to try to solve this problem.
I guess what I wanted to say was that in statistical physics, when you approach the critical temperature, you can define this reduced temperature, small t, that the temperature is scaled by the actual critical temperature, and then you not only see that many systems exceed the same critical exponents as you approach the transition, but also amplitudes that give you the coefficient.
Okay. Yeah. Okay. So I think there are a number of things to say here, and maybe the most basic thing to say is that our data are consistent with predicted scale, like critical exponents, but they're consistent with a rather large range of exponents there, and that's basically because measurements in biology are somewhat more noisy, but maybe even more relevantly, this is closer to like first order phase transition, whereas what people normally study are second order phase transitions, and that's really very relevant because if the second order is like a transcritical bifurcation, and what's nice there is that you can find exactly where that bifurcation point is because you can just tune it there and then you just locate it, and you don't have any of these extinction events, right? Because it's reversible. Whereas here, because you get the sudden drop, then it's not as easy because then some of them are going to go stochastically extinct, and so it's actually more difficult for us to locate precisely where the bifurcation point is. So, and that's very important for trying to measure exponents. Right? All right. So all of what I just said was talking about the dynamics in kind of a single isolated population. Right? But if you look at a natural population, they're extended over space. And so there's a natural question of whether there might be some sort of spatial indicators that some tipping point is approaching. And in this paper here, we collaborated with Kirill Korolev, a talented theorist, to demonstrate that there is a spatial analog to this classic recovery time. And the way that I like to think about it is that in the basic phenomenon of critical slowing down is saying that if this is population size, a function of time. If there's a perturbation, right, you might get a dip, and then you get recovery, and you can just measure that recovery time. And that recovery time will grow as the environment deteriorates as you approach this tipping point. However, I also showed you that even in the absence of a defined perturbation in time, even if the environmental quality is constant over time, you can look at the fluctuations in the size of the population to say something about how close you are to a tipping point. Right? And we measured an increase in the size timescale of these fluctuations. All right, so this is all looking at the dynamics as a function of time. But what about the dynamics as a function of position or location? Right, so here, if environmental quality is constant over space now, right, uniform environment, All right, then in principle you can look at density fluctuations, and this is something that that had been proposed in the field. All right, what was kind of surprising to us is that nobody had proposed or suggested what is kind of a natural fourth category of indicator here, which would be the spatial analog to the recovery time. All right, and so we're calling this recovery length, and the idea is that if you have a boundary between regions of different quality, right? So say that there's one region that that you fish and one region that you don't, right? Then just because you're in you're out of that fishing zone doesn't mean that you will immediately get to your equilibrium population density here. Instead, you have to get some distance away from the fishing region in order to get to your equilibrium density, right? And so there's some characteristic length scale, this recovery length, that just tells you how far you have to get away from a bad region, or for that matter, a good region, before you get back to your equilibrium, right? And what we did in this paper is we showed kind of theoretically, computationally, and experimentally in a simple model of our yeast populations that there is this increase in recovery length as one approaches the tipping point. And it's actually perhaps the easiest thing to measure of all four of these indicators, because you don't need a really high quality time series. You don't need to have a random defined perturbation in time. Instead, you just need to find a boundary between regions of different quality, which are actually kind of ubiquitous in nature, and then just measure the density profile across that boundary. And indeed, we have collaboration with a professor at University of Pisa, Alessandro Benedetti Cecchi. I always get confused about which is the C and which is the CH there. But and we have preliminary data seeing an increase in a recovery length in the context of a natural experimental community that is an algal canopy on an intertidal community. So it's a kind of an exciting opportunity for me to kind of get out in the field at some kind of real population, right? All right, are there any questions about this recovery length idea? Okay. All right, so that's maybe the bulk of the talk, but I'll kind of just give you a sense of some of the other kinds of ideas that we think about in the group. In particular, these ideas from evolutionary game theory. Right? So the populations that I've been talking about here were the same. These are our yeast populations, and let's just imagine they're all happily growing in some sugar, this sugar sucrose. And in particular, they're all secreting the enzyme. Okay? Well, then you always have to ask, well, what happens if one member of the population stops paying that cost associated with making and secreting the enzyme? Right? So if you want to, you could call that cell a cheater. You could also call it a non-producer if you'd like. Regardless of what you call it, it doesn't change the experimental results. Okay? All right, so so the question here is, if such a cheater were to arise, what is going to be its fate? Right? Now, if all the sugar, all the public good is shared among the population, then the only difference between them is that the cheater doesn't have to pay the cost to make that protein, so it actually will be able to spread throughout the population. And indeed, in the simplest models from game theory, what you expect is that that cheater cell should have an advantage relative to the cooperators at all frequencies in the population. And that's analogous to this idea of this prisoner's dilemma. And in that case, if it's always better to be a cheater, then at least extinction of cooperation in these simple, well-mixed environments where we're doing our experiments. Okay? Of course, we're experimentalists, so what we can do is we can try to measure these things. All right? What we do is we remove the enzyme, the gene required to make that enzyme, all right, the gene encoding the enzyme invertase. Right? And then we also label these two different cell types, indeed green and red, so that we can distinguish them by flow cytometry. Okay? All right, then what we can do is we can track the dynamics of that population as a function of time. All right, so the first thing is that you can ask, well, what happens if you take a population that's mostly cooperator and just add a few cheaters? Okay? And indeed, what we find is that the cooperator fraction as a function of time is decreasing. Okay? It's important to note that it's not that the cells are actually changing their strategy. These are genetically encoded strategies. So the cooperators give birth to cooperators, cheaters to cheaters, right? But rather it's just saying that the cheaters have higher fitness than the cooperators here. And indeed, in many of these conditions, what you see is that the mean fitness of the population here can actually decrease as a function of time. All right, so we normally think about evolution as a process that leads to always higher and higher fitness. And that is often the case, but when you have interactions between members of the population, these sorts of game interactions, then evolution can lead to decreases in fitness over time. Okay, and this is consistent with the basic idea of this dynamic being a prisoner's dilemma. And indeed, the reason that I got excited about studying this is because of a paper from Greg and Travasano showing that there was some game interaction in this yeast system and suggesting that it might be a prisoner's dilemma. Okay? However, when I went and I measured the cooperator fraction on the other side, when I just took a population mostly of cheaters and added a few cooperators, what I found is that the dynamics were very different, because now it's actually the cooperator cells that had the advantage. So what's happening here is that this is a situation with negative frequency dependent selection, right? So the rare strategies do better than the common strategies. And that's what leads to coexistence between these two strategies at equilibrium. Okay? And this phenomenon of coexistence is saying that we shouldn't be thinking about this interaction as this prisoner's dilemma, but it's really closer to what game theorists call a snowdrift game. And all these games have some story associated with them, and I'll tell you kind of the story of this one because it's relevant. So the idea is that if you're going home after a day of work and you get stuck behind a big pile of snow or a snowdrift, you'll get out and you'll shovel it away so you can get home, right? But if there's another driver next to you, then you're playing some sort of game, because what you would like is for him to get out and do the work of shoveling. Right? But if he's not going to do the work, then it's somehow similar to you being by yourself, so you'll get out and do the work. Right? So the key feature of this snowdrift game is that the optimal strategy is the opposite of what your opponent does. Right? It's advantageous for you to cooperate if only if your opponent is cheating, and then vice versa. Right? Now, this thing of coexistence is it's an experimental observation, but of course then you ask, well, why is it that the cooperators were able to survive in the presence of the cheaters? Why is it that it wasn't this prisoner's dilemma? And what we found was that it's because these yeast cells, they do indeed secrete the enzyme invertase that breaks down the sugar, but some of that enzyme gets stuck in the cell wall. Okay? And that's actually very important, because what that means is that some of the sucrose that gets broken down into the simple sugars that can be consumed, the public good is created locally. So those cooperative cells actually have some preferential access to the fruits of their labor. Okay? We found that preferential access was actually rather weak, in the sense that only about 1% of the sugar that was created by a cooperative cell can actually be captured or imported before it diffuses away and is consumed by other cells. However, that 1% preferential access is somehow essential to allow the cooperators to survive in the presence of the cheater cells. And actually, I think that this basic phenomenon is, I think, going to be very common in many cooperative systems. And indeed, we see similar dynamics in antibiotic resistance, where resistant cells, in many cases, break down the antibiotic. And in that case, those resistant cells can be thought of as cheaters, whereas sensitive cells can be thought of as sensitive cells would then be thought of as cheaters. I don't know if I said that correctly the first time. Right. And indeed, what we find is there's coexistence between resistant and sensitive bacteria in, for example, ampicillin. All right, so maybe here in the last 10 minutes or so, I'll tell you something about some of the population level consequences of cheaters in the population. Okay? And in particular, I think that there's an interesting way to think about this, which is something about coupling between evolutionary and ecological processes in these populations. Okay? Right, so we often assume that evolution and ecology are somehow not coupled, right, because of some sort of separation of timescales. Right, so the idea is that we think of evolution as being something that's very slow. And what that means is that if you want to try to understand population dynamics, changes in population size over time, for example, what happened on Easter Island, then we don't think about evolutionary explanations for why this happened, right? We look for ecological kind of explanations, right? And I think that that's maybe reasonable here. And of course, this is a bit of a straw man, because this is many, many, many mutations, right? But the idea is that recently, in, for example, Darwin's finches, there's been some studies showing that there actually can be coupling between evolutionary changes, for example, in beak shape and sizes of populations. And more generally, in microbial populations, selection is often very strong. And what that means is that evolutionary change can take place very rapidly, and then the two timescales may be more similar. All right, so to try to understand something about this, what my postdoc Alvaro Sanchez did is he repeated some of these experiments, but where he tracked both the size and the fraction over time. Right? And in the absence of any evolution, if you just have pure cooperators, then the population dynamics are relatively simple. Right? So I showed you that at the beginning of the talk, where there's some stable population size, right? There's a minimal size required for survival, otherwise they go extinct. Okay? But if you just do that exact same experiment, but now what you do is you have both cooperators and cheaters, then what you see is things are very complicated. Right, so if you look at the population density as a function of time, what you see is that some populations go up and then down, some go down and up, right? And it just looks very, very messy. Right? And things don't really get any simpler if you track the population composition as a function of time either. So if you look at the cooperative fraction as a function of time, then again, things go up, down, it's actually, you can't really make heads or tails of it. Okay? And incidentally, so that we're calling, this is evolutionary dynamics because it's a change in allele frequency as a function of time. Right? So the cooperators have an intact version of the gene, and the cheaters have a defective version. Okay. All right, so it's easy to look at this sort of data and kind of throw up your arms and give up, right? But that would be a mistake, because if you take this precise data and instead of plotting these quantities as a function of time, you instead plot them as a function of each other, okay, what you see is that you start to see some interesting patterns. Right? So here, this is population density on the x-axis and cooperative fraction on the y-axis. Okay? What you can see is that this red population is indeed going extinct, but these other populations are somehow spiraling in this space of population size and cooperative fraction. Okay? And what's happening is that here, we don't have very many cooperators, so they're not breaking down enough sugar, so the population size shrinks. But as that occurs, okay, the cooperators start to gain an advantage relative to the cheaters, so they spread. And as that occurs, now, okay, more and more sugar can get made, so now the population size starts to grow. Right? But once you get up here, this is the situation where the cheaters have a strong advantage. So now cooperator fraction shrinks, right? So we're somehow getting this spiraling in that space. And this spiraling is really a direct experimental manifestation of something that ecologists have been thinking about a lot recently, which is this eco-evolutionary feedback. So feedback between evolutionary processes and population dynamic processes. And this is neat because we can actually just see this directly in the laboratory, where each arrow here is just the change in these quantities over one day cycle. Okay. Indeed, we have a simple phenomenological model suggesting that, that maybe the dynamics look something like this. Okay, so if you're to the left of the separatrix, you should get extinction. To the right, you get survival, but spiraling to some state of coexistence. All of the dynamics that I told you about at the beginning of the talk were up here, where we had a pure cooperative fraction. So there's a stable fixed point, unstable, and again, stable down at 0. Okay, but what you see here is that actually the dynamics are much more complex than that, because we have this whole perhaps spiraling behavior. And indeed, when he did this experiment where he just started many different locations and then tracked the populations as a function of time, he saw that it really looks something like this. Our data over here are noisy because of the small population sizes, but indeed they all went extinct. Over here you see what are really, once again, kind of beautiful spirals to a state of coexistence. Okay. What's interesting here, I think, is that if you look at the size of the population in the presence of those cheaters, it's actually not significantly lower than the pure cooperative population, right? So it might be a 10% decrease in size of the population. However, there was a very strong effect, which is that there was a loss of resilience in the population as a result of those cheaters. Okay? Because resilience is still somehow the distance between the stable state and that unstable state. And you can see that the distance is much reduced in the presence of the cheaters. And we were able to confirm that there was a loss of resilience by perturbing the system and showing that in the presence of the cheaters, the populations could not withstand the perturbation. Okay? Now, it's interesting, and now that we have this slightly more complex population with 2 players, you can ask, well, how is it that these early warning indicators behave as we push this system to a point of collapse, right? And in particular, what we can do is we can look at these spirals, right? So this is, again, each segment is the change in the population size, composition over one day cycle. And they're really beautiful spirals, I think, to this state of coexistence, right? So this is saying now that the fixed point here is described not by real eigenvalues, but by complex eigenvalues. And the expectation here in the continuous kind of modeling is that the real part of those eigenvalues should go to 0. And so this corresponds to the spiral somehow becoming more spirally. All right, and indeed, in this paper, we've demonstrated we can measure that effect in these cooperative cheater populations. But I just want to show you what the trajectories look like in the context of the simulation, just so you can see what I mean by kind of more spirally. Right? So in a benign environment, you get rapid spiral to a state of coexistence. But as the environment deteriorates, the spirals take longer to kind of spiral in. Okay, and this is, of course, modeling in the absence of noise, right? And indeed, as the environment deteriorates, eventually you get to the point where that fixed point becomes, in this case, neutrally stable, right? So this, and then it's this point that you're going to get extinction and collapse of that population. Okay. All right, so this is a conference that's nominally about microbial communities, and these communities I've been telling you about are really very simple, right? There were all single species communities. In one case, there's maybe some different behaviors within that species, but much of what we and many other people are excited about moving towards is true like multi-species communities. And so, for example, our first paper looking at a coalition or a community of 6 soil microbes was just published last week. And there, what we wanted to do is just try to understand something about the repeatability of evolution. And this is something that people have studied in many contexts, molecular evolution or phenotypic evolution, but typically of a single species, right? And here what we wanted to do is ask the same question, but in the context of a multi-species community, where now we're looking at repeatability of coevolution, and we're looking, and the measurement is in abundance space, right? So asking about which of the species is doing better, or is occupying a larger population, let's say fraction of that community. So the idea is that in this 6 species community, there's some in some 6-dimensional space. there's coexistence of these guys, or at least coexistence of 5. Right? Now you can ask, well, after you do evolution, what happens? Right? So one thing that might happen is that you just get blurring in this space. Right? So just you get evolution, things kind of move around. Right? Or it could be that, oh, maybe all these communities all move to just some other location, right? Because of extreme determinism in the evolutionary process. Okay? But what we actually found was that it was really described by something more like this. So after 400 generations in this community, something like 20% of the communities went over here, and so species A did really well. In another fraction of the communities, species C did well. In another, so there was actually clustering in this abundance space after coevolution of these species. And I think this is an interesting thing for the theorists of you to kind of maybe think about what might be going on. And the last thing that we're doing in this multi-species realm is something that I'm rather excited about, which is using worms, C. elegans, as a sort of a biological model of island biogeography. Right? So the question there is, why is it that different islands are different? In this case, we're trying to ask, why is it that different gut communities are different? Right? So I have a different gut community than each of you. But of course, we're all different. We're different people, we eat different things, we live in different places. Right? But in the case of these worms, I think it's a really exciting opportunity where we can take identical worms, place them in identical environments, feed them identical bacterial communities, and what we nevertheless find is that there's sort of striking and really quite amazing differences in the gut communities of different individual worms. And I think we can actually make sense of this using some of these classic ideas from island biogeography. So if you're curious about these things, you should ask me over the next day. I am going to be here through the end of Friday, and then I have to go back to MIT to teach. So I encourage you, if you're curious about anything that I've been talking about, please ask me. All right. So I'll just summarize. So what we've been doing is using simple microbial populations to study kind of universal phenomena in the context of from theoretical ecology. In particular, trying to understand, in this case, what is the consequences of cooperative interactions with the population. Right, so in this case, we can measure things like a fold bifurcation leading to one of these sudden transitions. And we can also measure some of these general behaviors that are predicted from theory. So for example, we can measure an increase in the size and timescale fluctuations in the population before crossing one of these tipping points. We also found experimentally that in this system, at least, cheater strategies can indeed spread in the population, but that we get coexistence between cooperators and cheaters. And as I mentioned, this is something that we've seen in a number of other systems, so I think it's actually going to be quite common. Finally, we, I think, get a lot of insight by looking at these ecoevolutionary trajectories, where we look to see how evolution and ecology somehow feed back on one another. And I think that this is a neat opportunity to take interesting theoretical ideas and explore them in really quantitative ways with experimental microbial populations. With that, I think I'll just leave you with the acknowledgement slide, the students and the postdocs who have been doing all the work, as well as funding agencies which allow these students to get paid. All right, thank you very much. Yeah.
Would cheaters spontaneously arise in your tipping point experiments?
Yeah, so it's a good question. We haven't done exactly the experiments that would allow us to say definitively one way or the other. One thing that I can say is that we, in most of our experiments, we only allow them to go for 50, 60 generations, just so there's not enough time for new mutants to both appear and spread. But in some of that long-term deterioration experiment that I told you about, that was over many weeks. All right, so there's certainly going to be genetic heterogeneity in that population. So in that sense, it's actually quite encouraging that we were still able to see an increase in both the coefficient of variation and the autocorrelation before crossing the tipping point. Well, maybe you first, yeah.
In this measure of the collapse, this predictor, in the time measure, for the yeast system, you have some very, well-defined kind of, you can change the sucrose, you can change dilution. I'm just not clear, maybe this spatial thing solves it, but it's not clear to me, what if I don't know what to measure?
Okay, so I think this is an important question, and my claim, or the field's claim, is that that x-axis, in our case, we're imposing something in particular, right? But our claim is that the x-axis could be something that you choose, and you don't tell me what it is, but I'm still supposed to be able to anticipate that the tipping point is approaching, right? So it doesn't have, the x-axis, the environmental knob, doesn't have to be something that is well defined. And indeed, even the number, even for example, the sucrose concentration, that's varying a number of different microscopic parameters within the population, right? So it's not that it's a purely simple environmental knob. But the claim of the field, or the hope, is that even if it's something rather ill-defined, right? Even if multiple knobs are changing in different ways, then just the idea is that if you're approaching one of these tipping points, you're supposed to be able to anticipate that it's you're getting close.
But you have to be able to change something, though.
Well, no, we don't have to. It could just be the world that's changing it. So the idea is that global warming is increasing the temperature of the oceans, and then this is pushing a fishery towards collapse, right? So this could, so it's not that we are doing it. Well, we might be doing, but it's not that we're intentionally doing something, but rather that just the world is doing something, and then we just are measuring the properties of the population, and from that we're supposed to be able to, ideally we'd like to be able to anticipate that the tipping point is approaching.
Yeah, but I think this is my confusion maybe, that you have to know what. Suppose I have a virus that suddenly goes through my fishery at night and wipes everything out.
Yeah. Yeah. No, okay.
I have to.
Sure. Okay, but yeah, this is just highlighting that that it's always possible for a population to go extinct without any advance warning, right? So if a moon-sized meteor comes and hits the Earth, we're in trouble, and this type of early warning indicator is not going to tell you anything, right? So this is saying that if the environment changes somehow slowly, and the system is responding to that, then it should show up in the behavior of the system. But you can always have something that comes and wipes out a population without any advance warning, and that's just, I think, a fact.
A different time scale.
Yeah, right. I mean, it would have to be somehow system specific. You could maybe, if you're monitoring the viral population, then you might be able to say something, right? But that's a system specific kind of indicator you'd have to look at. Yeah.
There might also be other reasons for slowing down. For instance, if you lower the temperature, all the biological rates will also be slower, and the system will slow down, but it doesn't have to mean that you're closer to a tipping point or to any other modification.
Yeah, so I think that there are a bunch of things that can be said here. So one is that the environmental noise, I think, is maybe in some ways the biggest danger or so. In that the statistics of the noise can change, and that the noise could just increase in size, and then that'll maybe make the fluctuations in the size of your population grow, right? So I think that there are many things like that will lead to false positives, and of course you can also have false negatives. The particular example that you gave, I think that you have to be a little bit, some of the things that you might think of that are going to change it in this way might also actually increase the stability. So then maybe it is accurately reflecting. Although this is something that you'd have to. So it's certainly like a bead in a what do I want? Is this going to be true? All right. Since I'm being recorded, maybe I won't. Yeah. But yeah. So we'd have to think about exactly what perturbation or environmental change.
So you have to think carefully about your bifurcation parameter, which is the same comment as.
Yeah. Yes. That's okay. But then you have to look at it. You have to try to find a specific case that it really where it really is actually going to fail, and we can maybe try to figure that out over our sangria today at 5. We're going to go to tapas. Okay, but that's not for all of you outside of the world. But yes.
You said that when you put these worms, like in the last slide, when you put them in the same environment, they all have different gut communities. It's like really like that each worm has like an own fingerprint in the gut community, or is there like a finite number of different compositions of gut communities?
Okay, right. So these are rather preliminary results, but I think it's actually due to stochastic community dynamics. Right. So what we can do is we can actually switch, and just from island biogeography, if you have a large migration rate, then the island communities will each reflect the mainland community. Whereas if you have a low migration rate, then you have a lot of the equivalent of genetic drift, so like ecological drift. So and then you can get fixation of random things. And so we've actually been able to tune this migration rate by changing the feeding rate and show that we can switch between a regime where it's all the gut community is all a reflection of the outside, or if you randomly get one.
Okay, so some transition. So it's like at some point there's like a transition where the communities are completely random between different.
That's right. So it might be that one worm gets all this species, another worm gets all this one, and yeah.
But are they then changing over time, or are they for each individual worm?
Yeah, so right. So at least in principle, and we haven't fully confirmed this, in principle from the theory, you expect that each worm should, over long periods of time, stochastically move sample these different alternative, and they're not stable states, but should sample it. But it's over timescales that I think are longer than the 2-week lifetime of a single worm. So, but at least in principle, that's the expectation.
Okay.
Yeah, James.
So can
you extrapolate from measurements in the sort of stable phase of your curve to when you're going to see a collapse. So, for example, if you took your yeast system and looked at increasing temperature, could you measure the behavior under a regime where you can then predict at what temperature things are going to
All right, so my claim is that we would, and that's true whether we increase the temperature or we decrease the temperature, and we can argue about this later, because the time scale here that we're measuring, like the autocorrelation time, is over days. It's not the time scale, it's not the division time, or whatever, right? So, because these tipping points are a purely population level dynamical systems property, they're not a property of the individual cell. Right? So in our case, actually, the cells are getting happier as we increase the dilution rate because they never are saturating. Right? So just before this tipping point, they're actually always dividing and are happily going. Right? So I think you really have to think about the dynamics at the level of the population rather than the intrinsic dynamics, and that's why I think it gets more subtle than, you know. All right, maybe last question because it's going to be 3 o'clock in 20 seconds.
Yeah, do you later, no problem.
Okay, well with that, maybe we should quit so that we can get Alfonso going at the right time. All right, thank you.